Okay, how do we go from a messenger RNA, right, that we have a 5' cap and a poly A tail on, how do we go from that to an actual working protein in the cell? Okay, this is a process called translation. Translation happens in the cytoplasm. All proteins begin being translated in the cytoplasm. Okay, they all start being translated. They don't all finish there, but they all start there. Okay, so that's what we're going to kind of talk about today. How does translation work? And then how do cells that are staying in the cytoplasm, what happens to them? And then cells, what happens to proteins that are staying in the cytoplasm? What happens to proteins that actually have to move to a different organelle or have to be embedded in a membrane? What happens, or secreted from the cell? How do they move after they start translation? Okay? Everybody remembers the central dogma, which is what? Into protein. So DNA to RNA to protein. I can promise you I'm going to ask you that on the test. Okay? All right, so what do we need? So this is translation right here. Right, so we see the ribosomes in your book are always going to be the little yellow things. The protein, the polypeptide chain that's being made is in green, and the messenger RNA is in red. So what do we need for this process? It's pretty elaborate, actually. Okay, well, first of all, we have to have a messenger RNA, right? And this is a mature messenger RNA, right? So it has a 5' cap and it has a poly A tail, right? We got to have a ribosome. A ribosome, as you can see there, has two subunits. It has a large subunit and a small one. Okay, it also has 3 active sites inside that large subunit. Okay, so that's where the peptide bond is going to be formed, and the transfer RNAs are going to be moving through there. All right? To bring in the amino acids, we need another type of RNA. We need a transfer RNA. Okay, these guys. And there is one transfer RNA for every amino acid. Okay, at least one. Okay, the ribosome, I just want to mention now, has a ribosomal RNA, and it's inside the large subunit. And this is the enzyme that actually makes the peptide bond. Okay, so it is a ribozyme that forms that peptide bond. All right? To begin you have to have something that attaches the amino acid to the transfer RNA. Okay, that is another type of enzyme, these guys right here, called an amino acyl tRNA synthetase. It attaches that amino acid to the right transfer RNA. Okay, then when we start translation, we have some proteins that help assemble the ribosome and get the messenger RNA in the correct positioning. Those are called initiation factors. Okay, as the peptide synthesis goes on, we have other proteins that help do that. They're called elongation factors. And then when you reach that stop codon that we talked about, right, that's the stop sign, this is the end of the protein, you now have to kind of break the whole thing apart. So there are proteins called release factors that help disassemble the whole big complex. Okay, and then it can all be recycled to start something else. Okay, does that make sense? So let's take a look at ribosomes. Okay, so none of the pictures really show you the ribosomal RNA. But you cannot put the large and small subunit together without the ribosomal RNA, okay? And it is in what's called the P site. Okay, so I told you there are three kind of active sites within this ribosome. One is called the A site, the amino acyl site. That's where the transfer RNA comes in first. The one in the middle is called the peptidyl site. This is where you form a peptide bond. Okay, so this is like the start. And then the last site is the exit site. So this is going to be where once the transfer RNA has given its amino acid to the new peptide, it's released. Okay, so it's a channel where the transfer RNA can leave. All right, so you have them moving down this kind of assembly line. All right, and everything is added one amino acid at a time. You form one peptide bond at a time. Okay, and the messenger RNA is still going to go 5' to 3'. So it's going to be read in the ribosome 5' to 3'. Okay, so you can't just have it oriented in any direction. It has to be in that direction. So you have, and that's one of the reasons you have that 5' cap, because the 5' cap is where the messenger RNA actually binds to the small subunit. Now, everything starts on the small subunit and gets organized, and then the large subunit comes in. Okay?
Who? Okay.
And everybody remembers that
I need
codon
my bag.
is 3 base pairs, right, together.
So
Okay,
I don't know
when
if you can put something in the note if it's too late.
A peptide, to make a polypeptide, we're reading in sets of 3, right? And that is the reading frame. All right? So this is kind of
okay, yeah, because I'm going out tomorrow night. My buddies are coming
what is
in,
the start codon?
I don't want to worry about going to the bathroom.
Methionine, right?
And you don't remember how big the leg bags are, do you?
Okay, and the codon is AUG. Okay? So the reading frame is like reading.
That's fine. I was just gonna say I might have to just call Gabe or Max
where
and
you
tell them to meet me here in the middle of the day. Just
3 little words. Okay, so in this case, if you look here, we have kind of a lot of nonsense until you get down here, which says the big boy saw the man run. Okay, so in this case,
I'll be good until, but I would like to drain it before I go out.
Stand for the first codon, and you would read through that. Okay, it's very important that the reading frame is correct. Okay, if you have different mutations that can take place, so if you have a deletion, that's going to throw it off 1. If you have an insertion, that's going to throw it off 1. Okay, you could even have insertions where you have multiple base pairs put in, and deletions of the same flavor. You can have multiple base pairs taken out. That can be catastrophic because it can throw off the reading frame, and now you have just this nonsense. And while it might get some of it might get translated, probably the cell is just going to recognize it as garbage and destroy it. Okay, so now you could potentially be missing a very important protein. Okay, so if you take biology of cancer, that's one of the things we're talking about, I guess, in this lecture coming up. You know, there are lots of mutations that occur in cancers that are these insertions and deletions, where you'll have, different base pairs put in, or you'll have mutations happen so that these master regulator proteins don't work. And so that's how the cells kind of get out of control. Okay, and there are lots of different mechanisms behind that. We'll talk about mutations later on in the course. Okay? Does that seem okay? Everybody's good? Okay, so the 3 sites, the A site is the amino acyl site. It accepts the transfer RNA, so you'll have your transfer RNA with an amino acid, and it's going to come here. Okay, the P site is where the peptide bond forms. The E site is it's literally a channel, and that's how the transfer RNA actually is released. Okay, all of those are in the large subunit. Okay, the small subunit does not have any active sites. Okay, and that's because that ribozyme, the ribozyme is actually, that ribosomal RNA is sitting in the P site, okay, where it can catalyze that reaction to make the peptide bond. All right? So let's talk a little bit about transfer RNAs and the things that are associated with them. Okay? So this is a transfer RNA. Remember, we talked about RNA is single-stranded, and it has this kind of a unique ability to fold on itself, and it makes very elaborate shapes. The transfer RNA is a great example. This is kind of more realistic. So this is really more realistic of the 3-dimensional shape of a transfer RNA. Okay, just like any other nucleic acid, it has a 3' end, which is right here, and it has a 5' end, which is right here. Okay, I'm actually going to erase the 3' because it's a little bit hard to see. All right, at the 3' end, that is where the amino acid attaches. Okay, so if we had, let's say we had methionine, it would attach here, right? Kind of midway through the transfer RNA is another part that has what's called the anticodon. Okay, the anticodon is going to be complementary to the messenger RNA. Okay, so you're going to have your messenger RNA in the ribosome. The transfer RNA is going to come down. What's going to recognize that 3-letter codon or the 3-nucleotide codon is the anticodon. So it's going to bind there. Okay? So it's the anticodon that actually interacts with the messenger RNA. All right? So you have two ends of this, and you can see that RNA has these loops. So these places where that single strand is not complementary. That's how you get a loop. You get a loop when the base pairs are not able to pair. Okay, so you get these loops. Those loops that you see in these more elaborate RNAs are often sites where they're interacting with something, or you have a catalytic site in the case of a ribozyme. Okay? So this is the transfer RNA. We have a pool of transfer RNAs in the cytoplasm. We have a pool of amino acids in the cytoplasm. So this is very efficient. All right? So you have to attach that amino acid to the transfer RNA. It doesn't just happen spontaneously. Okay? This is a very specific type of reaction, right? We want the right amino acid attached to the right transfer RNA. So to do that, we need an enzyme, right? And those enzymes are these guys, the tRNA synthetases. They actually, you can see this is a really nice figure because it shows you that amino acyl tRNA synthetase in blue. And you can see how the transfer RNA in purple and the amino acid in green are bound in that active site. And you can see that the way it binds, this is the 3' end of the transfer RNA. This is the amino acid isoleucine. You can see in that active site, that 3' end where you need the amino acid added and the amino acid are right on top of each other. They're very close, okay? We'll talk about enzymes, I think, in the next chapter, but this is one of the ways enzymes catalyze reactions. They arrange everything so that the reaction takes place immediately. Okay, so they actually put the two things together so that the functional groups that are supposed to form the bond are right together, and the bond can form immediately. Okay, and this is nice because it also shows you that you have different amino acyl transferases per amino acid. Okay? Now, when you have an amino acid, like these guys, when you have a transfer RNA that is just free, it doesn't have an amino acid added to it, that is called an uncharged tRNA. Okay? When you have one that has that amino acid added, that is a charged tRNA. Okay? Yes. Well,
like you said, like it shows how each amino acid has there's different.
yes, they would, because so they have to have a different enzyme. That's a very good question, because remember when we looked at the amino acids, those R groups, they're all different. So they all have a different shape. So to get this reaction to happen, you've got to have a tRNA synthetase that has the right shape for every one of the amino acids. So yes, you have one for each one. Is that what you were asking? This question over here? No? Okay. And you can see also that at the anticodon loop, that also makes interactions with the tRNA synthetase. Okay, so that actually helps orient that tRNA synthetase in the active site. Okay? And we get these active sites by protein folding, which we talked about last time. All right? So this is kind of showing how you get the codon, which is in the messenger RNA, and the anticodon, which is on the transfer RNA, to come together. Okay? So this is our messenger RNA here, which we made in the nucleus. It's been transported now out into the cytoplasm. It has its 5' cap. It also has, it's not showing here, but a poly A tail, which I'll put over here. Okay, what's going to happen is, and it'll show this, I think, probably in the next slide or two. You have to have the messenger RNA is going to sit on that small subunit, and it's going to scan down that messenger RNA until it hits the AUG, right? Because this is the codon for methionine. Okay, so that indicates this is the start of the protein. This is where we should start reading 3 base pairs at a time. All right, and so you can see that here this is a charged transfer RNA with methionine. It can come into the A site and associate with that codon, right? And this is the anticodon, right? So it's complementary to that sequence. It's not the exact same as that sequence. Okay? And And so this is nice because it's
far
kind of
away
showing
from me.
you that pairing. Does that make sense? So this is how the process starts. If we look at the codons, so this is a table that shows you basically the genetic code. These are all the codons and the different amino acids that they code for. There are 64 total. Codons. 61 actually specify the 20 amino acids.
And I just went into Woods and Water for the first
And
one.
the other 3 are the ones you see in purple.
I'm gonna try and stay
Those
away
are
from
the
that
stop
place.
codons.
I went in to go.
Okay, so the way you would read this is
I
essentially
went
if you
in with the
have,
single
so
goal
say
of
we want
getting
to
a
find
belt,
methionine, we see A,
and almost spent like
U,
$500.
G. You start on this side,
There's just so
then
much
you move
shit
to this
that I
side,
want.
then you move to this side. So we have A, okay, and we have a U in the second position, so this is going to put us here. And we need to look down over here to G. Here's methionine. Okay, so that's how you read that chart. You'll notice that a lot of amino acids, in fact, the majority of them, have more than one
Yeah,
codon.
I've been trying to save money. I need to stay away from
Okay,
that
this is
place.
something called redundancy. And it is not just specific. We don't have just redundancy in the genetic code. We have a lot of redundancy in the cell in the important mechanisms. And this is because if a point mutation happens, and mutations happen at a pretty frequent rate in the cell over the course of an organism's lifetime, you still have, there's a little flexibility, especially in that third position, that third nucleotide. That's something called wobble. Okay, so it gives you some flexibility in case something happens. Okay, which is not unusual over the course of somebody's lifetime. The stop sequences have a couple of different names. Some people call them terminator codons. Some people say call them nonsense codons. So you'll hear all of those. I always call them stop codons. All right? Everybody good about codons? So translation has a couple of different steps. They're pretty straightforward. You have initiation, you have elongation, and you have termination. Okay, so we're going to focus on how this happens in eukaryotic cells, right? That's really the focus of our book.
What does it mean to repent?
So if we look at initiation, this is where you start to build the complex with the ribosome. All right, so the 5' cap on the messenger RNA is going to bind to the small subunit of the ribosome. Okay? And it's actually kind of held in there by the 2 proteins you see in kind of that pinkish purple. Those are initiation factors. Okay? Then those initiation factors and the ribosome itself are kind of going to scan down that messenger RNA and find the start site. So they're going to scan for AUG. Okay, so once you find the start, now what happens is you have the other parts of this complex recruited in. So everything is set. The messenger RNA is set. It's correct. You found that initial start. You found the start codon, and now the large subunit can come in and bind. Okay, so now you have this complex together to start translation, right? The first transfer RNA with methionine also comes in at the beginning.
Yeah.
Okay, so you have to have that there to kind of start. What are you going to add the peptide bond to? Okay, so that gets together. All of this gets together, ready to start in initiation. Once this is formed, those initiation factors can be released, and they'll go to start initiate translation somewhere else. Okay? The next step is now elongation. Read along this messenger RNA, form a new polypeptide. Okay? So this molecular machine is going to scan along. There's a channel actually that forms when you have the large and the small subunit together for the messenger RNA to kind of flow through. Okay? And as it reads along, it's kind of protruding out the back, if you want to think about it that way. All right? So we have our methionine.
Siri.
It scans down 3 nucleotides.
Take
The next
screenshot.
one is GUA, which codes for valine. So now we can have that charged transfer RNA come into the A site and bind. Okay? And once you have that, what happens is the amino acid in the P site forms a peptide bond to that new amino acid, the valine. Okay, so this begins, and so you're constantly going to just tack them on like that. Okay, and it's the ribosomal RNA that catalyzes the formation of this peptide bond within the ribosome. Okay, and then once you have this peptide bond, everything moves down. 1. Okay, so our first amino acid, this was the one that had methionine attached, is released through the exit channel. Okay, we have our growing polypeptide right here. In the P site, you can see in red, that's our new peptide bond, and the A site has moved down. Now we have a new codon, okay, that codes for this guy, arginine. So now this transfer RNA can come in. The anticodon and the codon bind, and there will be a new peptide bond formed between the valine and the arginine. Okay, and then everything shifts again. Okay, does that make sense? It's pretty straightforward. So again, the figure that the picture doesn't, the drawing doesn't show this, but there is also a channel that allows that growing peptide to just be extruded out. Okay, so this is a pretty big complex. There are a lot of channels for things to move in and out, because you could have, if we have a peptide that has 34,000 amino acids, that is a lot. So we're going to be extruding something that's really large. Okay, and so this is just showing you that movement. Now we have the arginine attached. Everything's going to shift over 1. We now can have a new charged transfer RNA come in with a new amino acid, and it's going to move all the way until it reaches that stop codon. Okay? And then when you get to the stop codon, which you see here as UAA, it's a little hard to see. Okay, once that is in the A site, what's actually recruited now is something called a release factor. So another protein comes in. Okay? It's going to enter the A site, and now this polypeptide, whatever it is going to be released. Okay? So that polypeptide is released into the cytoplasm. Okay, so All of this takes place in the cytoplasm. Everybody's good with that? Okay, so once it's released in the cytoplasm, a lot of proteins, they work in the cytoplasm, so they just fold. It is a spontaneous process. We don't know everything about it. We're still really actively studying how protein folding happens. It does not require energy. Okay, so there's no energy required for a protein to fold into a three-dimensional structure. All right, so this happens spontaneously, and all the information needed for folding, remember, is in that primary amino acid
Mom.
sequence. All right, so just to touch on prokaryotes, it's a lot simpler. We don't have these different compartments. Everything's happening in the cytoplasm, and it's not as, you don't have as many protein players coming in.
You know my weights, my wrist weights?
Make things happen. Basically, the ribosomes
Can you
are
put those out
assembled,
and remind you to bring?
and they actually
Remind yourself
bind to
to bring
something
those?
called a Shine-Delgarno sequence.
Okay.
Okay?
Yeah.
And at that point, they can
6.
start protein translation.
Yeah, so I'll probably leave my room at like
Okay.
1.
So the process is pretty similar,
It's gonna be a long day.
but it's a little bit
I got
simpler
a lot of-
because everything is
got a lot
just
of
taking
homework
place in
Sunday.
this one compartment. And of course,
I know.
only
And I feel bad
for- so I
bacteria
have
and archaea
a life group,
have
which is my,
Shine-Delgarno sequences. So eukaryotic cells do not have a Shine-Delgarno
which is my like Bible
sequence.
study group that I've been
We
going
have a
to.
much more complicated
I have to stop
promoter
going with them on
sequence and
Tuesday.
enhancer sequences that you don't find
Because I joined Ducks
in
Unlimited.
the microbes. All right.
And the meetings are at Tuesday at
You
6:30.
can see from this table and thinking about how many
No, I'm
amino
gonna
acids you
join
can
the
put
life
together.
group on
There's a
Monday.
huge number of possible proteins. And this was one of the reasons that
I know.
people
And eventually,
thought that
I'll have
proteins might
study groups
actually
with
be
fraternity brothers
the
instead.
information storage molecule of the cell
Yeah, I
instead of DNA.
I mean, I've only been going
Because
for a little
you
bit.
can make there's so many possibilities. And when we talk about regulation in a minute, we can also modify these amino
But,
acids. So there's even more.
yeah, I'm gonna start
potential
going on Mondays.
for different amino acids, different protein configurations. So there are a huge number of proteins compared to we have roughly 26,000 genes, but we make, I don't even know, hundreds of thousands of proteins. So it's, the gene sequence
Mama.
is not
see
reflective
me?
of the vast number
You
of
see me?
proteins we have in the cell that are working
See
to
my
keep our
genes
cells
now?
going.
No, look.
Right. Any questions about translation?
I went to the store earlier and got it.
All right. So all of these processes that we've talked about, transcription, processing of messenger RNAs,
Yeah, so,
movement in and out of the
Remind me to bring my
nucleus,
weights. Because I'm going to try and work out so I
translation,
can
all of
see you.
these are regulated very tightly. Okay? And we've already talked about the fact that not
I don't
every
have
gene
enough
is
strength.
expressed in every cell. Okay? Most
Because
of the
it's-
genome
they only
in
go
our
skeet
cell
shooting, like, once a year.
remains in what's called the silent state. It is wrapped up.
It's not a duck
coiled
hunting club.
into
A lot of
condensed form.
it's
not
Ducks
available.
Unlimited. Is
for
you know the license
transcription.
plate we have in the van?
Okay, so many
That's
genes
Ducks
are
Unlimited.
not
Pretty much we
accessible.
raise money. I'll let you know. I'm going to a meeting with. So Anna, Nurse Anna, how her sister cut my hair.
Yeah.
Good. So her sister was cutting another guy's hair, who is in Ducks Unlimited.
So they
And
can,
Anna was
we
like, I got
talked
someone
about
that's perfect
these
for
different
that.
superstructures that can form.
And,
You can form
Anna
chromatin,
just
you
gave
can
him
have
my
the
number, he
histone
just
kind of builds
texted
on
me.
a string complex. So
So,
every cell has its own kind of
so that's
or
one of
every
the clubs
tissue
I wanted
type
to
is going
join
to
while I
have
was
its own
here.
complement of genes that are being expressed, and some that are just They're constantly just kind of
They do
wrapped up,
duck.
and they're
It's
actually
pretty much just a hunting
relegated
club.
to the edge of the nucleus. Okay, if you look in an electron
No,
microscope, you
no,
can see
most of the
these
people have never
complex
done
of
duck.
DNA. It's just kind of shoved to
I'm
the
not.
side,
I'm not a duck guy.
so it's out of the way.
Only birds I like shooting are turkey.
Right. And we regulate
So
these processes.
I don't know.
Every,
I'm gonna go to the
talk
meeting,
about making a protein.
in a couple
Every
weeks,
step
so.
of the way, getting to the protein is regulated.
But,
so we regulate
all right, yeah. Remember my
transcription,
weights.
because we have all these transcription factors.
I'll
If
call
don't
you
have the
around.
Transcription factors, you don't get gene expression. We regulate RNA processing because there are splice variants. There
Why?
are small RNAs that can be used to regulate how messenger RNAs are actually made. And
They don't need
then,
to know.
even once a protein is made, we can
Oh.
still modify it. Okay, this modification takes place mostly in the Golgi. Okay, we can add sugars, we can add
Oh.
lipids,
Okay.
all kinds of stuff. You can add these modified amino
That's
acids. I mean, there are
pretty
all kinds of things can go on. Okay, so
Yeah,
even after a
it
protein
is.
is made,
Social
that
security
may
is.
not be the final
Okay, that makes
protein,
sense. Yeah.
because it may go
Let me know
through
how that
a
goes.
lot more processing.
Can you hear me?
Okay, so how does this work? How do we get proteins from kind of this, polypeptide in the cytoplasm? How do we get them folded and to the right place? Right? How do we do that? And that is a process called protein sorting. So all proteins begin translation in the cytoplasm. Okay, so
What you need? Wait, what? Wait, what do you mean?
okay, now some proteins are going to stay in the cytoplasm. Those are the easiest ones.
Uh-oh.
And some
That is
proteins are going to go through the secretory system.
How do you know it's wrong? In what?
Okay, so how can you tell the difference? How do you tell the difference between moving in these two directions? Okay, there is a special sequence
Okay.
that's added to a peptide that has to
Who
go
are you
into
with?
the secretory system. So it's
Oh, I thought I
called
heard
a
another
signal
dude.
sequence.
Oh, it's because I have my notes going on in the background while I'm recording them.
Okay, so those proteins have a signal sequence.
Uh-oh.
These guys have no signal
Wait.
sequence.
Just take another one tomorrow. I see the faint line.
Okay, so that's the first difference. If you don't have a signal sequence, you're a
Yeah, I know that.
and
the
that protein is
negative
just
blue line
going to
is
fold in
clear.
the cytoplasm into its three-dimensional shape. If it needs to be multi-subunit, it will join with other tertiary structures and form a quaternary three-dimensional shape, and then it will do its job. So pretty simple.
Hello?
Right? If you have this terminal sequence here, the signal sequence here, you're going to go to, you can go to the chloroplast, but we're going to really track to the mitochondria because we're mostly
Yeah, well, the
paying attention
negative.
on animal cells. Okay? There are
Those
some
things are pretty
proteins
accurate.
that have an
You took
internal
two?
signal sequence, okay, that you see here in C.
If you took two, I think you're good.
Now, these guys are special because
If
they
it's
are
just a faint
routed
line,
to
I don't
the
think that means
nucleus.
anything. And then, I don't know, give it another week.
Okay. So, the position of the signal sequence is important in where you're going to target a protein.
Hello?
Right? The most common is that you have a signal sequence at that amino terminal end, right at the end,
Oh.
and it's going to be targeted to the ER.
Yes. Yeah, why would
questions
you
about
tell him?
that? So,
How would it be his?
Because it's his. I know it is.
so if you remember, we talked
I
about
thought you said you never did it
the
wrong with him.
endomembrane system in chapter 2, right? So this is composed
Yeah, but I
of
thought you did wrong
the
with
rough
Duncan.
endoplasmic reticulum.
Yeah, that's
the
what I was thinking.
Golgi. We have
Well, it's one of those two, so.
vesicles. We also have vesicles here.
Take care of it.
You're a horrible person.
And the plasma membrane.
Duncan's a great kid.
Okay, so if you remember when we talked about the ER and the Golgi, they're these kind of
Yeah,
have all these
you gotta
folded
tell both of
kind of little compartments. So the ER has a space inside of it.
I'd tell Lane.
So the space right here is called the ER lumen.
Yeah, actually, that's a good point. Just wait.
And the
wait.
Golgi also has these luminal spaces.
Just wait.
Okay. The Golgi, if you remember, has an added feature, right? It has two different faces.
Yeah, just wait.
It has the face that's
No,
inside
no,
the
you're
space
good.
in the ER.
I'd wait to tell him.
This is called the cis Golgi.
Just try
And
and
it
close
has
your
the
eyes
side
and go to
that's
sleep.
facing the plasma membrane. This is called the trans Golgi.
Well, take another one. Do you want to just get a plan
Right?
B just
Everybody
in
remembers
case?
that from chapter 2.
It's not just like they.
Okay, so this is
I don't know how
where
they work.
those proteins that have that
I've never been in
signal
this
sequence
position
at
before.
the end are going to be targeted to. They're going to be targeted first to the rough endoplasmic reticulum. All right. So
I'd just wait a little bit.
once you have that signal sequence that comes out of the polypeptide
Yeah,
channel,
well, I'm not
in the
doing it. I'm waiting
ribosome,
for my
there's another
thing and stuff
protein
on.
that binds it.
And then I'm going
It's
to
called
put
the
the notes in the
SRP or the
AI
signal
thing.
recognition particle.
What happened?
Once that binds to
Shorter than
that
what's his name, Andrew?
signal peptide, translation
Oh, that's
stops.
hilarious.
Okay, so the
I'm
whole
like the same height as
ribosome
Andrew in my
with
chair.
the messenger RNA and everything just kind of freezes in
It's
place.
so funny.
Okay, then that whole
Yeah, if you're the
thing
same
is
height as me,
is
that's not
transported
good.
in the cell to the rough endoplasmic reticulum. Okay, and you can see here
I don't know.
there is a receptor
Hopefully you don't have a kid.
in the rough ER membrane,
God
and
damn
it
it.
recognizes the
I dropped my
signal
phone.
recognition particle. So these guys
I'll be right back. Can you hang up?
spines. Okay, this you can see
All right,
in the
bye.
next picture. This positions that ribosome over a protein channel. It's actually a tunnel, right? You can see that. And now what's going to happen is you're actually going to have this part of the peptide released from the signal recognition particle, and it moves into this channel, and the ribosome then associates with that protein channel. Okay, once that's set up, translation starts again. Okay, but now that peptide is being extruded into the ER because it's going through that protein channel. So this is the ER lumen. Okay, so it's going to go until you reach the stop codon. Okay, when it reaches the stop codon, you're going to have the polypeptide terminated, and then it's going to be released into the ER lumen. The ribosome and everything is going to, break apart from that channel, and it can start over again. Okay, does that make sense? So that's how you would get a protein to enter this whole system, going through the rough ER to the Golgi, and then finally packaged in a vesicle to either be transported, secreted out of the cell, or transported to another organelle, whatever its target is. Okay? Does that make sense? So this is just showing you this anchoring, okay, and the new polypeptide being synthesized and secreted into the ER lumen. The ER lumen has a lot of enzymes, and one of them actually cuts off that signal sequence, okay, because the signal sequence is not part of the mature protein. Its job is to get the polypeptide that's being translated, the whole complex, to the ER. Once it gets it there and translation starts again, it's done its job. So it's just really, it's degraded. Okay? Does that make sense? So we have these proteins in the ER lumen. And so let me go back here. So the protein you see here that has been completely released into the ER lumen, it's going to go somewhere, probably targeted to an organelle, or it may be secreted out, but it's not going to be embedded in a membrane. Okay? A protein that has to be embedded in the membrane has stretches of hydrophobic amino acids in it that actually are going to be embedded in the membrane. And they also have to enter the secretory system. Okay, so if we have a transmembrane protein, remember a transmembrane protein is a protein that goes through the entire plasma membrane. It has an extracellular side and an intracellular side. Okay? If a protein is going to be in the membrane, you can see this kind of purple here. That is a stretch of hydrophobic amino acids. You can't put those into the ER lumen. That would be a big flashing, red light. Those actually, what happens is this channel is actually very elaborate. It can open on the side. And what it does is it actually moves that hydrophobic stretch that's been translated into the membrane. Okay, so what you end up with is a transmembrane protein. Right? It has the hydrophobic part embedded in the hydrophobic part of the membrane. It has a C-terminal region facing out into the cytoplasm. And it has an N-terminal region that is in the ER lumen. Okay? So in this case, what's going to happen is as these are made, you have these proteins that are sitting in the ER membrane. They actually form vesicles, and then they're transported out. Some of them might be going to the chloroplast, the nucleus, the mitochondria, to be embedded in those membranes. Or they could be headed for the plasma membrane to renew the plasma membrane. Okay, so the plasma membrane, because things are usually constantly budding off, it loses a little bit every time a vesicle buds off. So you have to replenish that. Okay, so as we said and talked about in chapter 3, the membrane is a very dynamic place.
Ducks Unlimited.
Okay?
I joined
Does that make
Ducks
sense?
Unlimited.
So
I joined Ducks Unlimited, and the meetings are every
So proteins evolve just like everything else.
other Tuesday.
We have about
At Mode's at
25,000
6:30.
different protein families. We group
Are you a part of
proteins
that?
really by their sequence and by different structural elements.
I love Jesus. He's so.
And we do this because if you see these common sequences that make these common, larger structures when they're folded, it gives you an idea about what the function is. And so you have so all of the tRNA synthetases, they're all different, but they all share a lot of similarities because they're doing the same job. Okay, so we have lots of proteins that do the same job, and we kind of group them into these families. And lots of people who are biochemists and biologists spend their whole lives studying, one or two proteins within a group. In my lab, we study a type of protein called E3 ligases. And these are the proteins that they target other proteins for destruction. It's a really
I would
important
never
way that
give
we
it up for
regulate
anything.
a lot of stress processes in our cell. My lab studies innate immunity in plants. So, there's a lot of study of these different proteins. Okay. We talked about protein folding. We talked about alpha helix and beta pleated sheet. So you can see here, these are two examples. What the protein that's on the left is alpha helix and beta pleated sheet. And then the protein on the right is something called a beta barrel, which you probably are learning about in chemistry. This is a shape that a lot of membrane channels have, because you have the beta pleated sheets kind of embedded in the membrane, and then there's a channel going through. So you can have a lot of things moving in both directions, actually. And the loops that you see in gray, that's where things can associate on both sides to be channeled back and forth. Okay? So if we look at the number of folding domains, there are a lot fewer of those, right? There are only about
Yeah, I go
2,500
2 or 3 times a
folding
week.
domains. And that's just kind of how it is. All right.
But I just joined
Just because you have
one
you
of
can
the
have many
clubs,
different folding domains within the same protein.
a club that
Okay, so
has
some proteins are
meetings
very large, so you have a lot of room for a lot of
on
different
the same
things
day.
to form. And so we talked
And it's
about
my-
we haven't talked about dehydrogenases yet, but we'll talk
Life
about those
group.
when
So I'm
we
either gonna have to
talk
switch
about energy
days.
production and respiration. So dehydrogenases are huge proteins, and they have 3
That sucks.
different enzymes inside of them, each one with a totally different job. And they're channels that are moving things from place to place, things coming in, things going out. So each part of that protein has, that overall protein, the dehydrogenase, has many folding domains within it. Okay? And we also kind of look at proteins. These folding domains give us information.
ever have
When we
time, I
find
can
an
add you to
unknown
the
protein,
group
or we have a
meeting.
protein that has an unknown function, you can look at the different domains.
One of the
and
guys found me
that
on
gives you an idea.
Instagram
Well, this might be
because
doing this, and
he
then
gets his
that leads to
haircut.
ways you could test. Does this protein have that
Hannah's
function?
sister got me in
Okay.
touch with one of the guys.
So a lot of evolution, which you'll see as we kind of go on, happens through mutation. And so there's some mutation. It can be large or small. It could be a point mutation where only one nucleotide is changed. It could be something really catastrophic where you have a whole piece of a chromosome that gets deleted. That happens. But sometimes when these things happen, it's not bad. It actually ends up creating something that gives that organism a survival advantage over the rest of the population. And so in that case, that's going to be conserved, because in most cases that individual of whatever population it is going to be able to survive longer, better, produce more offspring. So now this mutated or new version of whatever this protein is going to be the majority of the population. Okay, so things move forward like that. So if something is positive, it has to have a survival advantage. Nothing is going to be conserved over evolutionary time in the genome if it doesn't provide an advantage to the organism. If it's not providing an advantage, that's a problem. There are some things that can be neutral. Most of the time, these are actually gene duplications. So if you look at our genome over billions of years, sometimes genes just get copied multiple times, and they're typically sitting right next to each other. We don't really understand why that happens, but it is a way that you can evolve the genome. Okay? Negative, if you have a negative mutation, it typically takes away from a survival advantage. It causes the organism to be less fit. We'll talk about
That's fucking
fitness
crazy.
populations later on. And so that individual probably would not reach reproductive age or would not produce progeny. So now this would just take it out of the gene pool.
Hello?
Okay? Any questions about that? So this chapter has two really nice, has I think there's 4 really nice big figures looking at transcription overall, RNA processing overall. So this is going to be right in the nucleus. So, and then it also has transport out of the nucleus and translation in the cytoplasm. So I'm a visual learner. I learn a little better like that. I like to look at pictures, and this is a very nice tool. I think you should take. It's not for this exam, but for next exam, I think it will be helpful. Okay. All right. Please take advantage of Kelsey's session tonight. I will see you all on Tuesday.